Okay, like Tim O'Brien said for the Lake Huron, this is a combined presentation with our bottom trawl survey and our acoustic and midwater trawl survey. And so we have two reports outside, and the co-authors of all those reports are named here, and these are the agencies that contributed to these data. So just to give you a sense of the complementarity of these surveys, and I think as we go through this talk, you'll get a sense that they really are telling us similar stories about the prey fish biomass despite some of their differences. So the bottom trawl survey goes back to 1973, except for one hiccup in 1998, it's been continuous. The acoustic survey has been continuous since 2001, also has 5 years of data in the 1990s. Of course, they occur at different times of the day, the bottom trawl during the day, acoustic at night. They vary with the number of trawls that are conducted. They also vary in terms of what part of the water column is being sampled, right? So the bottom trawl survey is sampling about 1.5 meters off the top of the bottom of the lake, whereas the acoustic survey has a dead zone about where that bottom trawl survey is sampling, and then there's another dead zone about 2 meters below the surface, because the echo sounder essentially is pointing downward. So they're not able to catch the fish that might be above that echo sounder. The bathymetry is also different. Since 1973, the bottom trawl survey has sampled between 9 and 110 meters consistently at about 10-meter intervals within that. And Brian Breiter alluded to this earlier. The acoustic survey is not a fixed survey like the bottom trawl survey. It goes out. And you can see this is you saw this earlier, that's the 2014 map of where the transects were located. And then they have 3 species that are common, alewife, bloater, rainbow smelt, and I'll show you estimates for both of those. And then the bottom trawl survey has a set of species that is unique only to that survey. So again, just to emphasize this point that Brian made, just showing you how these acoustic transects vary from year to year. And this is the same plot that Brian showed, showing the lake-wide coverage out into deeper waters and again, moving those transects from year to year. So today I want to step you through, first of all, talking about the species that are common to both surveys, those species that are unique to the bottom trawl survey, and then some of the things we've been thinking about with respect to the design of these surveys, thinking about some of the biases that are inherent in any survey, including these, and then finally some take-home messages. So the common species. And so what you're going to see for these common species are plots like this, where we've relativized both of our time series, focusing only on the years in which we have common years for both of the surveys. So 5 years in the 90s, and then all the years since 2001. And so what we've done then, for example, this is the peak biomass for large alewife. In the acoustic survey, we've divided each year of the survey by that number, so you can put these on the same scale. This is the one that probably looks the least similar between the ones I'll step you through, and that's because the acoustic time series was saying that the peak was actually in 2002. Sorry, the bottom trawl survey, the black is bottom trawl. Bottom trawl is showing a peak in 2002, whereas the acoustic survey said there were more large alewife back in the 1990s. But one commonality, of course, is the 2014 data point being quite low for both surveys. Looking then at just the acoustic, so now I'm showing you kilograms per hectare with the error bars, again for large alewife. Again, you see higher estimates in the 90s, lower levels in 2012, including 2014. Now the bottom trawl time series, which dates back to 1973, so everything to your left of that green point here is essentially saying this is information that that I haven't shown you in earlier graphs, and you can see that buildup of alewife biomass back into the 1970s, and when Chuck Medengian extends this back to the 1960s, the biomass was even higher. So analyses of these time series has indicated that the primary driver for alewife in Lake Michigan is salmonine predation, with secondary effects of warm springs and warm summers also leading to stronger recruitment, which in turn leads to larger populations of alewife. When we look at the age distribution of the alewife population based on the bottom trawl survey, in yellow you see the majority of that adult alewife is the 2010 year class, about 40%, and then showing up smaller contributions from the 2011 and 2012 year classes. Of note, we are not seeing any alewife larger than 180 millimeters. Another way of thinking about whether or not this alewife population is truncated is, let me step you through this graph. In blue here, this is the percent of the alewife population that is age 6 or older in our bottom trawl time series. So you can see that before 2007, it was not unusual to get measurable percentages of age 6 and older alewife, some years more than others. But since 2007, that percentage is almost zero consistently. In the background there, you're seeing the maximum age sampled by the bottom trawl survey, and you can see again before 2007 that number bounced around between 6 and 9, but again since 2007, very consistently a much lower maximum age, bouncing between 4 and 5, and then 1 year of 6 there. When we look at the small alewife, these are best indexed by the acoustic survey, based on where they live in the water column. And you can see that this acoustic survey has picked up 3 strong year classes, the 95, the 2005, and the 2010. Had the survey been out in 1998, you would have seen probably an estimate way up here somewhere. It would have been probably off the charts. Now shifting to bloater, our second common species. This is one that shows quite high correspondence between the two surveys. Peak biomass in the 90s, much lower biomass in the 2000s. And again, very low levels in 2014. Looking now just at the acoustic time series, showing you kilograms per hectare with the error bars. Again, another thing Dave has done is sort of pulled out the noise you see in here, and you can see actually there was a significant increase in bloater biomass in 2014 relative to 2013. Shifting to the bottom trawl time series, again, looking at the data earlier than 1992, you get a little bit different picture. A huge buildup of bloater biomass in the 1980s and 1990s that Steve Podhoven just referred to, and very low levels of bloater, large bloater since then. Looking at small bloater, again, fairly strong correspondence between the two time series. Very low levels of recruitment of small bloater in the 90s, and then some evidence of production of small bloaters in the 2000s. Again, this is the same time series for the acoustic estimates. A little bit different perspective when you look at the bottom trawl time series, however. This is so before we were looking at low levels in the 90s, increases in the 2000s, sort of sporadically, but that pales in comparison to what we saw back in the 1980s that drove that huge biomass of bloater over that decade or so. So when we look at time series to try to explain the variation in bloater recruitment, there's really no single one factor that we can say is the primary factor. We found a couple of contributing factors, including the reduction in fecundity in the 2000s. Bloater are less fecund now at similar sizes than they were back in the 1960s and 1970s. We attribute that to the loss of diporia and their reduced condition. We completed a study recently that looked for the evidence of slimy sculpin egg predation, as well as egg predation by deepwater sculpins and round gobies. We did find that in a few years in the 2000s that predation could have been significant enough to dampen recruitment success, but it certainly did not explain the variation we saw could not explain the variation across that entire time series. The last species then, large rainbow smelt, again, strong correspondence between the two surveys, much higher densities in the 90s than in the 2000s. Again, now just looking at the acoustic time series with the error bars, giving you a sense of much higher kilograms per hectare in the 1990s. Now with the bottom trawl time series, you see that, again, in the 1980s was a period of much higher biomass of rainbow smelt than we've seen since 1992. Looking at small rainbow smelt, again, pretty strong correspondence between the two surveys. Lower levels of recruitment, or intermediate levels of recruitment in the 90s, more variable levels of recruitment in the 2000s, some really strong years of strong smelt production. Again, now just looking at the acoustic time series, and now the bottom trawl time series, we see, again, here's that noise, the intermittent high levels of small smelt that were produced in the 2000s, still much smaller than what we saw back in the 1980s. Zach Feiner from Purdue University has recently analyzed this time series and found that actually a stock recruit model, the best stock recruit model for this particular time series was one that actually split it into two time series. So one before 2000 and one 2000 and later. And what he found when he fit a stock recruit model in these in these later years is that the productivity of that stock, that is the number of recruits produced per adult, was much higher in the 2000s than it was back in the 80s and 90s. So they're still able to produce these young, but those young are not surviving to larger sizes. Okay, so let's then move on to time series of species that are unique to the bottom trawl time series. The first is round goby. We've heard a lot about round goby at this meeting. Admittedly, we are not sampling round goby in their ideal habitat, which would be harder substrates, shallower waters than what we're sampling. But we do, so even in the sort of suboptimal habitat where we're sampling, though, we think we're still capturing some of the trend of an increase in round goby biomass followed by a reduction in more recent years, likely associated with predation, as more and more of these predators are taking advantage of this abundant prey. Deepwater sculpin is our next species. Deepwater sculpin had a recovery in the 1980s and 1990s, in large part due to reductions in alewife. Alewife would prey upon the larvae of Deepwater sculpin, based on some work done by Chuck Medengen. Burbot is another species that is that can have a negative impact on Deepwater sculpin because burbot will consume them as prey. In more recent years, we think we're not actually sampling, we're missing a lot of the Deepwater sculpin population. Our survey has stayed at 110 meters, 9 and 110 meters, and we think that a lot of the Deepwater sculpin have shifted into deeper waters that we are not sampling. Interestingly, Brian Weidel at the Lake Ontario Committee presentation showed that they've been going out and doing deeper and deeper tows in Lake Ontario and found that Deepwater sculpin biomass continued to increase even out to 210 meters in depth. So again, I'm not sure that this is a real decline in Deepwater sculpin biomass. It's just an artifact of our survey. Deepwater sculpin is not an artifact of our survey design. We think that we're sampling almost all of the population between 9 and 110 meters. When Chuck analyzed this time series through 2002, he found that juvenile lake trout was the primary driver negatively influencing slimy sculpin biomass. Since 2009, we've seen slimy sculpins decline to record low levels, and about the only hypothesis that makes sense is that this decline coincided with the increase in lake trout spawning. So you have more juvenile lake trouts out there, and they could be contributing to this decline through predation. Nine-spine stickleback is one that we don't hear a lot about, but actually over about a decade period they got up to about 1 kilogram per hectare of biomass, which is quite high for a fish that's very small. This is comparable to what we're seeing in some of the years for round goby. Not to say that there's as many sticklebacks as round goby, but again, there were a lot of sticklebacks out there for a while. So one potential explanation for their increase over this period was actually a beneficiary of Dreissenid mussels, and in turn the proliferation of Cladophora. And there's a recent paper published by the Michigan Tech Research Institute which used remote sensing and found that in fact the rise in Cladophora quite perfectly matches this rise in 9-spine stickleback, which tickled Chuck because it confirmed his hypothesis. And what we don't know, however, is why sticklebacks are much lower now. A0 yellow perch is another species that we can index with our bottom trawl survey. We're able to capture that increase in perch recruitment in the 80s and a few strong year classes over the past 10 years or so. Sadly, we didn't catch any A0 yellow perch in this year's survey. And finally, Burbot. This is a species that recovered quite nicely in the 80s and 1990s as a result of control of sea lamprey and then also reductions in alewife biomass over the 60s and 70s that allow for their increase. And again, this is a alewife preying upon little burbot larvae. It seems that the burbot population is now settled into lower levels than what it was able to maintain in the 1990s. So I just want to talk briefly about some of the things we're thinking about with respect to these surveys, both the bottom trawl and the acoustic. I've referred to them a little bit throughout the talk. The first one is how is water clarity affecting catchability for this survey that happens during the day? And so you can see this is a time series provided by EPA. This is spring Secchi depth, so how deep can you see that Secchi disc as it goes down in the water? And you can see that since mussels have come in, the water is getting clearer and clearer. So we don't have great data to try to get at this question, but we have some data from 2012 in Lake Huron using the same net we use in Lake Michigan that where we were sampling during the day and then at night within a 12-hour period. And for a species like a Deepwater sculpin or a round goby, one that we don't expect is undergoing diel vertical migrations on the bottom during the day, up at night, this might be something where we can try to get at this catchability issue. So here are the data for Deepwater sculpin. So what you're looking at here, this is different seasons and sites within Lake Huron, Hammond Bay, Thunder Bay, at different depths, 46 meters and 82 meters. The white bars are day trawls. The night bars are, or the darker bars are trawls, 12 hours later. And there's no pattern. We see no differences between day and night that are consistent that would suggest any sort of catchability issues. That's not the case, however, with round gobies. You can see that we are, especially at 46 meters, we are getting more gobies at night than during the day in our bottom trawl. Now, just to sort of confirm our hunches that these fish are behaving as we think, when we look at catches of rainbow smelt day versus night, we're getting significantly higher catches of rainbow smelt during the day. At night, of course, we think this is indicating that they're up in the water column. And again, the same thing for bloater, day versus night. So these don't really help us in terms of the catchability issue, but they do confirm that these fish are performing behaviorally as we expect. So there's inconclusive evidence so far in this sort of exploration of net effective catchability. It's going to take future work to try to understand this. The second thing for the bottom trawl survey is this notion that the depth distribution is changing for some of our fish, in particular Deepwater sculpin, but also for bloater. There have been a lot of reports from commercial fishermen that bloater are captured much deeper over the past couple of years than they were even a decade before. And so the way we've tried to get at this question is through some opportunistic deeper trawling. So when we have time in the day, we'll go out. When we end at a 110 meter site, we'll go out to a 130 meter site, and we've been able to do this 5 times. Again, I'm just going to show you data for Deepwater sculpin and for bloater. So for Deepwater sculpin, the data were much like we expected. 3 out of the 5 times we're actually seeing the highest catches. So this is depth, and this is the grams per hectare. This is biomass on the y-axis. So most of the time, in fact, we're getting more biomass at deeper waters. I'm not sure what's happening at Port Washington in the Wisconsin waters, both years. For bloater, we're seeing some evidence of this only at 1 of the 5 sites, however. And so again, it's still possible that bloater are deeper than even 130 meters. We also have data from the acoustic survey. These are data that Dave Warner put together, which does go out deeper than 130 meters, even out to 260. And what they found at night with the acoustic survey is that the bulk of the bloater population is in waters 110 meters and shallower. So moving forward, we'll continue to do opportunistic deeper trawling, and then to try to get at sort of the potential water clarity issue. We hope to add a light meter to try to see if there's any relationship between water clarity and then some of the catches that we're getting in our trawl. The Midwater Trawl Acoustic Group has been much more active in thinking about the potential biases of their survey and some of these catchability issues. They've been submitting proposals. They have research underway. Some of the things they're actively working on is, again, trying to look at that upper blind zone that I described. They also are thinking about what's the effect of the vessel and the noise of the vessel on causing fish to get out of the way of the sounder. This is something that's much more difficult to estimate. Thinking about catchability, not only in terms of are the fish there, but to what extent are the fish going inside the net, or are they getting out of the net once they're in the net? And so moving forward, in fact, this year, at least USGS hopes to implement micromesh gill nets to try to and set those up in the water column to try to get at that sort of upper dead zone. Also would like to deploy upward-looking transducers on something like a glider. Actually, I may not have that right. I guess you would tow that. Talk to Dave if you want more information about this. The glider would be something, essentially put a sounder on a glider and compare that. How do the acoustic estimates differ between a vessel and something much more quiet? And then also, I think working with Gene Adams, there's a project underway that's trying to simulate what effects catchability could be having. So briefly, because I know I stand between you and the social, a few take-home messages. Prey fish biomass is at a record low. This is the plot that, I don't know how long it's been put together for the bottom trawl time series, but we continue to do it. This year we also put it together for the acoustic time series. And you know, these surveys that are taking place different months, different times of the day, sampling different fish communities, you know, biomass is much lower than it used to be in the 1990s. And again, just another way of looking at this, relativizing the two time series, really low biomass in 2014. So what might be the cause of this? From my point of view, I think it's clear that most people know that there's a strong top-down effect, especially on something like alewife. Chuck's papers have documented this in terms of effects on alewife recruitment. Eob Sahay's paper demonstrated this quite elegantly with the statistical catch-at-age models in the paper published last year. So a clear top-down effect. But also there is more and more evidence that there could be also some bottom-up effects, effectively squeezing prey fish. So reductions in nutrient inputs, and this is long-term since the Great Lakes Water Quality Agreement. The proliferation of drycenids that essentially reduce the efficiency of the food web. If we have all of this energy bound up in tissues and shells of mussels that perhaps otherwise might have gone into zooplankton or into phytoplankton. We have the loss of species like Diaperia. We have reductions in, this is a little cartoon Daphnia. All of these things, although we don't have clear evidence of this in terms of evidence on fish abundance, there is some suggestion that they're reducing the condition of fish and potentially their biomass. Secondly, again, another interesting place where these two surveys converged. About less than 20% of the prey fish community in Lake Michigan is comprised of native species. And then lastly, I'll just leave you with round goby. This is a species that was not at a record low. It's also a species that we admit that our survey is not designed to sample most efficiently. So I encourage you to stick around tomorrow. Randy Claremont will be talking about this issue, and he's put together a couple of different data sets, I think, that are and a couple of different ways of sampling gobies that I think you'll really enjoy to help us better understand and come to a better lake-wide estimate of brown goby biomass. So I'll just end by thanking again the vessel crew of the 4 outstanding vessels that allow these surveys to be completed each year. Thank you.